Good morning to all. Today we are going to discuss observation of cell under the microscope. How we can observe the cell under the microscope. So here we are going to observe the cell, dead cells, plant cell and animal cell. Plant cell and animal cell we are going to observe under the microscope. So we are going to observe dead cell, plant cell and animal cell. For that we required some of the materials we required. First one we required the compound microscope. First we required compound microscope and again we need a glass slide. We required and cover slip. It is nothing. Glass slide we required and again we required the cover slip. It will be helping to cover the material which we are placed on the slide. It is very small and thin in structure. It is not a thick and all. It is very thin in structure. And after that we required a water bottle. These are the things we required, and we required some of the stains also. Stain. What is the stain mean? Stain is nothing but it will be helping to coloring the cell. If you are adding some of the stain to the cell or the material which we are going to observe under the microscope, we can observe the structure of cells detailing. Without the stain, if we are observing, we can't be separate the cell structures or cell organelles which are present in the cell. We can't be separate. We can see some of the group of something is present, that much only we can see. But the structure of the cell, clearly if we need to observe means, definitely we need to add some of the stain. Clear? And here, first we are going to discuss going to observe dead cell. For observing the dead cell, what are the materials we required? Otherwise, what are the object we are going to take? You can take one matchstick. Matchstick is available now in your home. It will be very much familiar for you, right? We all daily we are using. So matchstick, you can take one of the matchstick from that, and you can take one small piece, make it transparent, very small thin structure, otherwise thin part we can remove from the matchstick, and you can soak it in a water for few hours, and after soaking you can take some part from it. You can soak the matchstick for water for few hours, and after soaking you can take and place to the microscope. You can place to the glass slide. You can, and after you can add one drop of water, and after that you can cover with the cover slip and observe under the microscope. You can see like a small room, so you can observe under the microscope. Some small room like structure you can observe under the microscope. This is the observation of dead cell. Next we can discuss, otherwise we can observe the plant cell. For observing the plant cell, we require the same materials, but instead of matchstick we are using the onion. Okay, you can take the onion. Matchstick, that is the dead one, but here we are taking the onion. Clear? So you can take one onion and cut into the small pieces. You can take one small pieces from the onion. One small piece you can cut from the onion. While cutting this one, by cutting and if you are separating, we can break the piece of the onion. You can remove one transparent layer. One transparent layer you can easily remove. You can take the transparent layer and you can place the transparent layer to the glass slide. But without making any folding, you can place the transparent layer to the center of the slide. After that, you can place it, and after that by using the needle or anything, you can place it properly without any of the foldings. Don't make any foldings. And after that you can add, we need to observe the nucleus, otherwise the structures no. So that's the reason we can add some stain like methanyl blue. So if you need to add some stain, that stain is known as methylene blue. Methylene blue we can use. It will be helping to observe the structure of the cell clearly and neatly. Otherwise we can observe it clearly. So for that we can add a stain known as methylene blue. We are adding to the structure which are placed on the slide. After that we can place a cover slip, and you can add one drop of glycerin to avoid the dryness of the cell. So for adding, you can add one drop of glycerin. After that you can observe under the microscope. You can observe under the microscope. You can see one structure like this. While observing under the microscope, you can observe a structure like this. This is nothing but it is the plant cell you are observing under the microscope. Here you can see, by observing this you can understood the outer covering, that is the cell wall is present in the plant cell. The outer covering is the cell wall, and in the center of the region you can see one structure, that is the nucleus. And in between this nucleus, cell wall, otherwise cell membrane and the nucleus, some space will be there, that there will be filled with a jelly like substances, that will be known as cytoplasm. That will be known as cytoplasm. Now clear about this all the things. While you are observing the onion, you can observe under the microscope, you can get the structure like this. That will be having the cell wall, nucleus and cytoplasm. These are the structure of the cell. You can visible clearly. Is it okay? And next we can going to discuss another cell we are going to observe under the microscope, that is the animal cell. So for the animal cell, what we are going to observe? The human cheek cell. So in our body, in our mouth, this beside your mouth, they having some small cells. The cells will be present. That epithelial cells we are going to remove from your, by using a spoon or spatula. You can remove the cheek cell from your mouth and you can place it on the slide. You can place it on the slide. You can spread it properly. After that, the same process what you are done for the observation of onion, that the adding that you can continue here. Methylene blue you can add, and after that you can cover with the cover slip, remove the excess stain. After you are observe under the microscope, after you can observe under the microscope. But here the cells are very separate, but the size shape of the cells will be different here. The cells, they will be, because by spreading here the cells, different cells are forming. That's the reason we are observing under the microscope the cells which will be present separately. So here the center of the region, nucleus is present. The outer covering, cell membrane, covering cell membrane and cytoplasm. These are the structures are present while observing the animal cell, the human cheek cell under the microscope. Is it clear now for you, the observation of cells under the microscope? Okay.